I'm Professor Greiner in the Department of Computing Science. It's my pleasure today to welcome you all and to moderate this meeting. Let me begin with a land acknowledgement. The University of Alberta acknowledges that we are located on Treaty 6 territory and respects the histories, languages, and cultures of the First Nations, Métis, Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. I also want to thank Paladin for the support of this ATI seminar series. Today's presentation will be an artificial intelligence-driven, multimodal computational pathology. It'll be given by Dr. Mahmood. Quick bio, he's an associate professor at Harvard Medical School and the Division of Computational Pathology at the Brigham and Women's Hospital. He received his PhD at Brigham and Women's Hospital. He received his PhD in biomedical imaging from the Okinawa Institute of Science and Technology, Japan, and was a postdoctoral fellow at the Department of Biomedical Engineering at Johns Hopkins University. His research interests include pathology image analysis, morphological feature, and biomarker discovery using data fusion and multimodal analysis. Dr. Mahmood is a full member of the Dana-Farber Cancer Institute and Harvard Cancer Center, and also an associate member of the Broad Institute of Harvard and MIT, and a member of the Harvard Bioinformatics and Integrative Genomics faculty. And now, if he's ready, he can begin his presentation.
Perfect. Okay, thank you so much for the introduction, and it's a pleasure to speak here today. I'll be talking about some of the work that my group has been doing in computational pathology, and I know that this is a cardiac seminar series, but a lot of the work that my lab does is related to cancer, and a lot of the methods that we have developed are related to cancer. So I'll be talking about some examples in cancer, but they're mostly to set up the stage and to describe the methods that we built that then led to the study that was of interest to your group. So just to get started, I thought I'd say a few words about computational pathology in general and what my lab is very interested in doing and what the field in general is focused on. So we analyze a lot of phenotypic, morphologic level data, and it's typically a lot of analysis on H&E data, where we're doing a lot of quantitative spatial analysis with the hope of correlating the quantified properties with everything here in the red box, so including early diagnosis, prognosis, prediction of response to treatment, patient stratification, identifying new, more innovative biomarkers. But another thing that we like to do is that we like to include markers that are already quantitative and are increasingly becoming available, and this includes genomic, transcriptomic information, and so forth. And we use a lot of modern deep learning and machine learning methods and algorithms to integrate this information together. And a lot of times, what my group really does, and we're a group of about 20 graduate students and postdocs that come from a variety of different orthogonal backgrounds, ranging from computer science and bioinformatics to people who have gone through entire clinical training and our postdocs. We try to build methods that enables doing all of this easier and more accessible. So for today's talk, I thought that because a lot of our work is focused on cancer, I'll talk a little bit more about the methods, and some takeaways from this talk would be that how can you build weakly supervised deep learning methods for pathology applications. So using pathology images and making predictive determinations from pathology images in an easy and efficient way. The second takeaway would be what are easy approaches to integrate data from multiple sources using deep learning to essentially get to everything in that red box or here. And then finally, finding similar pathology cases using deep learning. And I'll talk a little bit about what applications can this have and why we are interested and excited about this area in general. So to give a small sort of historical perspective about this, about the field of computational pathology. Computational pathology involves analysis of of pathology slides that have been digitized and scanned, and an increasing number of medical centers around the country and elsewhere are increasingly digitizing their pathology slides. So you end up with an image that essentially looks like this, but this image is very large. It can be 100,000 by 100,000 pixels, and it has a wealth of information. So historically, a lot of this data has been analyzed by using some form of handcrafted feature engineering, where we're based on human knowledge. We would look at information within the image and model that information using mathematics essentially, but using other well-known computational tools and features that are commonly used in computer vision, or have been used in computer vision. And then there will be some kind of a learning task towards the end. About 6-7 years ago, when convolutional neural networks became very common, a common approach has been that someone goes in and annotates the whole slide image, and based on that annotation, we would say, okay, this region is morphology X, and everything else is morphology Y, and can we classify them? And we used a deep neural network, which does not require us to manually handcraft or engineer any of the features. An issue with this approach is that it requires these manual annotations, which can be slow and difficult to obtain, and requires expert annotation. It's a little bit far away from the way these cases are essentially collected clinically. A solution to this approach is the utility of weakly supervised deep learning methods. And these are much closer to how this data is collected, used, and analyzed for clinical in a clinical setting. So clinically, whole slide images or a series of whole slide images would get assigned a diagnosis, which would reside in a pathology report. So if we were to use these very large gigapixel images and just the label that's in the report, that would make our lives as computer scientists working on this much easier, because they would not require annotations, but they would also open up a whole range of applications that these methods could be applied to at scale. So here's an example. We have a whole slide image. There are two ways to do this. So the example is a whole slide image. We can segment out all the nuclei in the image, build a graph on top of it, and then use something like graph convolutional networks that would learn what the most important nodes are and make a slide level prediction. The other approach is to use something called multiple instance learning, which assumes that this large slide is a bag of smaller, tiny patches, and we can represent each patch as a lower dimensional representation. And this encoder could be trained on just using real world images, or it could be something that's slightly more fancy, like a self-supervised encoder, but encoding these patches into a lower dimensional representation and learning from those. This, as I said, enables us to solve a lot of problems that are large scale, yet using data and information that is already collected for clinical purposes. So just to take a step back and reiterate some of what I already said in a more descriptive manner. So this is a whole slide. It's very large. It can be 1 billion pixels. It's typically analyzed using a microscope. It's just a glass slide analyzed under a microscope. Now it's been digitized and it's very large. So a typical workflow for most deep learning studies has been that someone would go in and annotate a region of interest within this slide, and then eventually you'll have a region of interest that's annotated by an expert. And you can chop it up into finer, smaller patches, and then those patches can have labels that would then be fed into a convolutional neural network, and you would train this. And the question that we were very interested in answering when we got started with this in this field was that, can we use whole slide images and just slide level labels? And the answer is yes, but the issue, the key problem or challenge here is that each one of these gigapixel whole slide images becomes a single data point. And that's a lot of data corresponding a single label. So it's essentially computational pathology in general is like a very sparse label sparse labeled and low data regime. A solution to this that people have used commonly is that you can have this very large image, chop it up into smaller patches, have these patches, and then use the label or the case level label that was available in the pathology report and assign it to each and every patch in the image. And then the issue with this approach would be that there would be a lot of noisy labels in the patches that you would get from this. And this approach could work quite well for larger sections where large portions of the slide correspond to the label that's assigned. However, if you look at regions which are very small, which might have a very small region that we're trying to identify, this approach would essentially fail. The other approach is to use vanilla multiple instance learning, where we use whole slide images and slide level labels, assuming that each slide is a bag of smaller patches, and even if a single patch is positive, the entire slide would be positive. They use something called max pooling based aggregation functions. And this essentially means that when you train your deep model, your model can be updated using just a single patch. So you're essentially using a single patch to update your deep model from this very, very large image, which makes it inherently data inefficient. There was a study published in 2019 in Nature Medicine that originated from from MSKCC in New York City, showing that we needed around 10,000 cases to get 10,000 cases to achieve performance that would be at the level that would be clinical grade at the level of a pathologist. Now, the issue here is that it's not possible for us to obtain this many cases for a majority of problems that we would want to target. And the question really boils down to that: is there a data efficient way for us to do this? And if we are able to answer this question and have a method that would be data efficient, yet being weakly supervised, that would allow us to do things by using data that's collected for clinical purposes, yet being data efficient, and would allow us to target and solve a lot of critical problems. So, for example, rare conditions, clinical trials, where it might be interesting to predict which patients would respond to treatment and patient stratification, things for which you would not have a lot of labels and data available to begin with. So, as a solution to this, our group developed what we now call clustering constrained attention multiple instance learning. This was a study that was published in 2021. And what we did here was that we tried to address a number of key issues to make multiple instance learning more data efficient. And we used attention-based pooling, which means that the aggregation function to generate a slide level computational representation, we learned what the most important patches are in the whole slide image. We also use n-parallel attention branches in some cases, which allows us to solve multiple multi-class problems. Pathology in general is inherently multi-class. There can be many findings on a single slide. We use something called pseudo labels. This allows us to enhance data efficiency. And we also use pre-trained encoders, which means that we use AI models trained on real-world images and use the first few layers of those models, and we're able to extract features from them to get representations of pathology images. And this is akin to how humans essentially see the world, and these earlier layers often learn very basic image features like edges and corners and things that humans would use to represent vision and image around the things around us. So this is how the method works. We have a whole slide image. We can segment out the tissue region within the image. Then we can patch regions within the image so to get to smaller patches that are much more trackable. We can extract feature representations from these images. Once we have those feature representations, we can address downstream we can solve essentially the downstream problem by having an attention backbone that learns what the most important patches are, as well as having multiple branches that would look at multiple classes, eventually going to a slide level prediction. We have some other bells and whistles that are built in there, such as instance level clustering, which clusters together similar features, allowing the method to be slightly more data efficient, as well as attention pooling, which pools the most important features within the whole slide image. Because we have attention built in, this means that the model is interpretable to a degree, and we're able to go back and look at what the most important regions were within the whole slide image. So the initial study targeted 3 cancer-related tasks. One of them was breast cancer lymph node metastases. When breast cancer metastasizes, lymph nodes are the most common location where it essentially travels to. This was done because it's a commonly used benchmark in computational pathology. It's a slightly more difficult problem than just detection or subtyping. It was trained on a publicly available data set and adapted to data from the Brigham and Women's Hospital. But I think the key takeaway here of the analysis is the data efficiency of the method. As we reduce the number of slides that were used and required used for training from about 700 to just 100, the performance dropped, but the performance was still relatively reasonable. And then how the performance essentially decreased for when we adapted the model trained on external data to our in-house data sets, as well as analyzing the regions that the model was using to make classification determinations, and both qualitatively as well as quantitatively, being able to say that the regions the model uses is corresponding what would typically be used in diagnosing some of these cases. We also wanted to push and see how far can we take this in terms of domain adaptation. Can we train these whole slide images and adapt to real-world images that are just taken with a cell phone coupled to a microscope? And we did some analysis around that and found that while there is a drop in performance, the model was still adaptable. And looking at what regions the model used in making those determinations, they were corresponding what a pathologist would typically use. So one thing I want to mention about this pipeline and method before I jump on to another application is that this code base is publicly available, and there's been a growing community of people who have been using this method. Another application we targeted is a particularly difficult problem. It's for cancers of unknown primary. When cancer metastasizes, it's often unclear where it may have originated. And we did a study where we used quite a lot of cases. And the question we really wanted to answer was that, is it possible for us to use conventional H&E images to identify what the origin of the tumor is? 22,000 cases were used to train this model into 18 different classes. We used internal and external test cohorts, as well as the cohort of cancers of unknown primary cases that came from multiple external medical centers. And the key point to highlight here is that some cancers are more common than others, and this is a generic problem. So we had very few data points, very few whole slide images and slide level labels for some of these cancers, and a lot of them for others. And this is also a problem that we would not have been able to solve if we did not use a data efficient method and a weakly supervised methods, because we cannot possibly get annotations for over 22,000 cases. It would be a very, very difficult task. The architecture used here was weakly supervised multitask. So this means that we were asking the model whether the case is primary or metastatic, as well as what the origin is. And then we did quite a lot of analysis on the binary problem of predicting primary versus metastatic, as well as predicting what the origin is and analyzing each possible origin, as well as looking at the high attention regions that model used in making those classification determinations for all 18 of the cancer types that were studied, as well as quantifying everything in the high attention region to be able to say that, quantitatively say that we're fairly confident that the model uses the cancer regions in making those predictions. So this brings us to the study that I think is more of interest to your group, is is related to endomyocardial biopsy assessment. So I think no one here potentially needs an introduction to this, but if there are people here who don't know, after heart transplants, patients will often undergo repeated heart biopsies to determine if the donor heart is being rejected. And this problem was interesting. This was the second problem we targeted after we had built the method, is because it came up repeatedly in talking to people in our department and elsewhere, that this is not an easy problem for, it's one of the difficult problems in pathology, because there's a lot of inter and intra-observer variability between pathologists and even between the same pathologist evaluating cases again. And the criteria has changed several times over the past few, or the past 15 years, I believe. And we wanted to see that, is there a way for us to use these computational tools to help standardize this a little bit better than the way it is right now? And that led to this the study that was published earlier this year. So our study design was relatively quite simple for this. We used about 1,300 biopsies from over 4,000 whole slide images, 1,300 biopsies from the Brigham and Women's Hospital. These were scanned at 40x magnification using a Hamamatsu scanner. And they were adapted to, the model was built and trained on this data set. They were adapted to and tested on an internal cohort of about over 300 biopsies, as well as 2 external cohorts, one that was sent over from a medical center in Turkey. And it used a different slide scanner, different staining protocols and everything. And another one that was sent over from Switzerland using a third slide scanner and very different staining protocols as well. So we had the opportunity to investigate the adaptability of these models in quite some detail. The model architecture that was used was very similar to the Clam-based architecture that I showed in the last 2 examples that I presented. So we have a whole slide image, we segment and patch it into smaller regions. Then we extract features from these regions. These features are then, we learn the attention, and that's where it was split into 3 different tasks, because these 3 morphologies of whether a case has cellular rejection, antibody-mediated rejection, or quilty lesions, which are benign mimickers of rejection. These 3 morphologies could exist simultaneously on the H&E image. So we wanted to make those predictions simultaneously given one single slide. And then there was a separate network that handled high versus low grade. So we grouped the grade 1 and 2 and 3 were grouped together. And then there was a third network that separated 2 and 3, and I'll talk a little bit more about why that was the case. So here is the model performance. Is the performance on the held out test set that was not seen by the model during training on cases from the Brigham and Women's Hospital, as well as cases that were sent in from Turkey and Switzerland. As you can see, that there's a drop in performance when we go from internal cases to external cases, both in terms of the AUC as well as in the accuracy. And a lot of it could be attributed to the difference between the cohorts and how the data was collected and prepared. So in the case of the US cohort, we used a Hamamatsu scanner, whereas for Turkey, it was a Aperio scanner, Leica Aperio scanner, and for Switzerland, a 3D Histex scanner was used. The slide staining protocols were also very different across the 3 cohorts. And just examining the hue saturation also shows us that how the 3 cohorts are different. And for the purposes of investigating the adaptability of the model, we did not use any form of domain adaptation here. We also were able to investigate in quite some detail what the model uses, what morphologic regions in the whole slide image the model uses in making those classification determinations. And for a majority of cases, we found that the the regions that are typically used were determined by the model on its own without using any pixel level annotations. So we trained the model just on the whole slide images and what was available in the pathology reports. And the model was able to identify these common regions across the set classes on essentially on its own. As well as investigating how these heat maps look like when we are adapting to data that was sent over from very different medical centers, and just investigating it in close detail. So the initial, if we go back a little bit to the architecture, the basic network could tell you whether a case had cellular morphology, antibody morphology, or quilty lesions, and a separate weakly supervised network could tell you whether the case was high grade versus low grade, and here's the performance. But there were very few cases available for grade 3, and separating grade 2 and 3 became sort of a difficult task for weakly supervised learning. So we used pixel level annotations on these slides, and here's the performance for slide level performance, while the model was trained on pixel level annotations for separating grade 2 and 3. Another thing that we did, and this was a really small study. This requires, we would need to do this at a very large scale to develop a more concrete answer for how much the model actually helps. But in this particular case, we did some comparison with human experts. The typical way this is done for a lot of the cancer studies is that there's a consensus that's developed and the models are evaluated against a consensus. In this case, one thing we found that the cases where, and this is very similar to what other people have found in other studies, is that for this particular task, the cases where a consensus could clearly be developed were relatively very easy cases. So we took a sort of an alternative approach that has also been used in some other AI studies, where we compared the performance between 5 experts, and here's a plot of their performance based on Cohen's kappa, which is also taking into account agreement by chance, and then also compared their performance against an AI model, and we can see that there's some performance improvement here. But I think perhaps this one sort of clarifies it a little bit more. This is another experiment, again done with five experts, where they were randomized to see whether they look at whole slide images and whole slide images with AI assistance for 150 biopsies, and after a washout period, they look at the reverse. And then we could see that the performance for detecting, so when they look at just whole slide images, it's just whole slide images in the demo that you can access over here. And when they look at plus AI assistance, they essentially look at the heat map and the whole slide image, raw whole slide image as well. So the performance improves for detecting everything, basically from cellular, antibody, quilty, and grade, and the assessment time decreases. Now, I should mention here that these are like very, very small studies, and these should not be taken for concrete answers. For the AI would help when we do this in a very large setting, in the setting of a larger study or a clinical trial. But these are indicators that such investigation is would be worthy to do. The next few things I'll talk about are again going to be more cancer related, but it might help you sort of understand a little bit where the field of digital and computational pathology is going, and how could you use these tools for your own research. So I'll present this study that was recently published in Cancer Cell, where we're trying to integrate information from histology whole slide images as well as molecular profiles. In this case, we use both mutations from a NGS panel, as well as transcriptomic information. So the goal here is that, can we fuse this information to build better prognostic models? And can we discover new prognostic markers doing so? So we use the whole slide image, and this is very similar to a Clam-based setup that I showed earlier. And this is a self-normalizing network. These two deep networks are fused towards the end, followed by 2 fully connected layers. So these are trained independently, followed by fully connected layers, and trying to rank the patients, and then being able to plot something like this, where we can show how separable cohort is. But the interesting aspect here is that we basically just feed in the whole slide image and the entire molecular profile. What does the model use from the whole slide image and the molecular profile? And can we quantify it to identify new markers? And can it go beyond what we already know about these cases? So that's perhaps the more interesting aspect. So we did this for 14 different cancer types, and we found that I think 10 of them were separable in a statistically significant manner. And we were also able to investigate for which cancer types was it beneficial to use the whole slide image and for which ones was it not. I mean, you could sort of pose this in a number of different ways. You could say, is it enough to just use the histology whole slide image to prognosticate few cancer types? For some, that might be the case. And for which ones would it be beneficial to have both histology and genomics included? When combining histology and genomics, we found that patients were more separable for a majority of cancer types, but then looking at the regions that the model used in making those determinations and being able to quantify everything in the tissue microarchitecture and being able to separate it showed us a number of different things. And I'll refer you to the article for some more details. Here's another example for melanoma, where we're trying to separate these patients, and it does better when using both histology and genomics combined. One interesting aspect that we found was that because the model uses all of the data and we do not quantify the data individually before feeding it into the model on its own uses regions of interest that it sees fit for prognostic determination. So one interesting aspect that we found that the model on its own picked out tumor infiltrating lymphocytes, and that's aligned with a growing. So it's very, very, very aligned with the growing hypothesis, and it's basically been proven using a number of different techniques now that tumors that elicit a strong immune response don't generally do better, and the model sort of identifies it on its own. And there are some demos and other information available if people are interested in exploring this further or even using the method for other applications. More recently, we've extended this further by including histology whole slide images, radiology, and genomic information, and then fusing all three of them together to see if prognostic profiles could be even better. And that is the case. And I think that a lot of the deep learning methods for combining information are going to be very important in multimodal data integration in the next few years, and would find a lot of applications in both integrating for better prognostic diagnostic determinations, but also for identifying new markers, new integrative markers for a variety of different disease models. And the last thing I'll talk about is this very interesting study that recently came out in Nature BME, and we basically wanted to see if we can build an effective search engine for histology whole slide images that would be very easy to use and would essentially not scale. The speeds would not scale with the size of the repository that it searches through. So I think it's worth mentioning a bit of a history here. So I think historically these glass slides are typically stored in large repositories, and it's impossible to find cases that might have similar morphology. One could look through the pathology reports and find and essentially use search tools for pathology reports to find descriptions of morphologies or similar cases with the same diagnosis, but they're perhaps not as descriptive enough as actual morphology can be. So identifying cases with a similar morphology in large cohorts of data is of significant interest, and it could have a number of different applications. Finding cases with similar morphologies opens up AI for rare conditions, because we never have enough cases available to train conventional AI models for some of these rare conditions. It also opens up the possibility of doing something with historical repositories. If you have a large historical repository with millions of slides where you don't have electronic pathology medical records, it's almost impossible to use them. But using something like this, you might be able to identify lots of similar cases with with ease. And then other questions that, do patients with similar morphologic representations have a similar response to certain therapeutic agents? These are things that we don't yet know about, but is definitely worth exploring. So the study was recently published, and I'll go through the method very quickly. We have a whole slide image, and we build a mosaic representation from the whole slide image by using very simple k-means clustering to cluster similar morphologic regions. So instead of using the whole slide image, we can use this mosaic. And then we used a pre-trained encoder, and this encoder was trained on using a vector quantized variational autoencoder, where we have the image, we encode it into a feature representation, and then build back the image. And in the end, we use this learned codebook. So we extract features given patches using this encoder, as well as an encoder that's just trained on real world images. And we have a random Bose tree, which allows us to do very fast search through these cases, and then an uncertainty ranking based algorithm that allows us to go from those patches to similar slides in a large whole slide image repository. So given a slide like this, you could find similar cases like this, as well as investigate what regions within the slide were used to make those essentially similarity determinations. And there was quite a lot of analysis that was done on this for a variety of different cancer types. It's generally applicable to any disease model, but a lot of our analysis focused on cancers using large public data sets such as the TCGA, as well as our own internal data sets, and then investigating how the model performs when the number of cases that the model is searching through are slowly essentially increased. So I'll stop here to allow for enough time for questions, and I'd like to thank everyone who's currently working in the lab, as well as all the funding that we have received to do some of this work.
So you talked about finding similar cases, but what about creating large language models as have been used for GPT-3 and DALL-E 2 and so on? Yeah. Radiology has gotten fairly deeply into that. Yeah. There is one resource, Pathology BERT, but as far as I can tell, that's the only one so far. Wouldn't that be the most exciting future thing to do? And bring, pathology up to the same level as these other areas of fast-moving science?
Yeah. So that we're trying to do that. So we have a number of people we're working on trying to build a large language model together with images. There are some very unique, as with the conventional learning and classification and everything, there are some very unique challenges to pathology. So one such challenge is that, of course, we can build something like Pathology BERT fairly easily because we have access to millions of pathology reports. In fact, we have trained something like that for pathology reports from MGH, from over 1 million reports from MGH. The challenge is with paired image and text data set that you would need for the next step after that, right? So the text model can it can be used for a few things, but it can't be used for much unless you have paired image and text information. Right. Now, the challenge here is that the pairs for radiology and the pairs for pathology are very different. For pathology, the relationship is incredibly weak. So if you have a whole slide image, you could have a tumor in there, and you could have normal tissue, and you could have a lot going on. Whereas pathology, the images are relatively small. So we're trying to address those challenges by
but it's high stakes, and it's quite important. And what it would allow, and this is something that would be astonishing for people, you put in data and you get a whole slide image at the other end, right? I mean that's really way beyond anything that we're doing. At the recent Banff 30th anniversary pathology meeting, I predicted that you can't do this with, DALL-E 2 type functionality, because it requires knowing the relationship between various parts of the image and like tubules, glomeruli, and so on. Really, DALL-E 2 isn't capable of keeping track of stuff like that. But DALL-E 3 and 4 will be, and that will exist in a year or two. So I was telling people by the time of the 2024 Banff meeting, you'll be able to input text and get a whole slide image. So I'm counting on you to do something like that.
So yeah, I think one of my PhD students was there at the Banff meeting, and I heard that there's a lot of interest in using AI for renal transplant.
It's obviously possible. Let's make it happen.
We have an ongoing project with doing similar to what we did for cardiac transplants. Of course, renal is a slightly more difficult task, but we're working on that too.
Great. No, that's a wonderful talk. Thank you.
Dr. West, all right.
Hello, that was really interesting. Thank you so much for that presentation. This might be a really simple question and betrays my relative lack of experience with these methods, but in the setting of transplantation, do you find that the cellular complexity of the organ itself further adds to the ability to discriminate these, these states of rejection when it manifests differently from liver to heart to kidney, and also the complexity of the organ itself?
Yeah, so in general, we don't translate them across organs, right? So a heart-specific model would stay a heart-specific model. But my general observation after working in this area for quite some time, and I worked on not just pathology images, but the spectrum of medical data using deep learning algorithms, is that every disease model there's in any kind of image, there are essentially two things, right? So there are patient-specific features that are specific to the patient, and there are diagnostic features that are common across patients, right? And the diversity of patient-specific features and the diversity of diagnostic features often sort of determines how good of a model, how robust of a model can you build, right? Some things like cancer, the diagnostic features are very reproducible, whereas for a transplant we've seen that the distribution is a little bit more broad, which makes it slightly more difficult to build some of these models.
Thank you. Yeah, it's very interesting to see how these will evolve, as Kim says, through the next couple of years. Really powerful technology.
Let me ask a question, if I may. So again, fascinating talk, lots of questions and comments. One of your one of the penultimate topics was involved survival prediction, survival analysis, where you built Kaplan-Meier curves for different subpopulations. Have you thought of building individual models so you can tell, for this patient, give an estimate how long that patient will live afterwards, or time until progression or something?
Yeah, so we've looked into those. Those are possible. This all depends on the amount of data that we have available for these. The issue with a lot of the multimodal setups is that it's very difficult to have enough data available to build these models. But for some of the common cancer types, I think it has been shown that you could do prognostication as well as some of the commercially available assays using genomics and so forth, just from the histology image itself.
I'll send you a point to some of our papers on that topic. Other questions? Maybe I'll ask just another one, if I may. There's a lot of talk right now about variational autoencoders, or VAEs, as defined intermediate low dimensional representation or submanifold. You sort of hint at something like that, but do you actually explore VAEs, and did they not work, or this work better?
They work. They're a form of self-supervised learning,
Yes.
right? So you essentially define your ground truth from within your data, and there are a number of different ways to do that. Atom coders is one of them, but there are a number of other ways too. I think given enough data and enough diversity in the data, going to a lower, training a very rich feature encoder that can encode your images into a lower dimensional feature representation has a lot of value, because you can do a lot with it, right? Like the image search I showed is one example, but you could do, you could train models more in a more data efficient manner. Or just makes training easier, right? So if you don't need to train end to end, you're able to train it with those features. Yeah.
Cool. Other comments or questions? Well, maybe I'll ask one final one. You showed the disappointingly large cap or small kappa values between pathologists and certain tasks. Are there ways to avoid needing human interpretation? For example, going right from the slides all the way to predicting the downstream outcome, and use that as the label as opposed to stage 2 or something like that?
Yeah, so I think that is possible. It's often made a little bit more complicated because of comorbidities and other issues that are that can come. I mean, just the causality of the when the data was collected, what else was going on. So we are looking into that, and I agree. One thing that's worth mentioning here is that while there can be a lot of intra and intra-observer variability, the deep learning is very good at dealing with noisy labels, right? So the way that it essentially it's based on feature learning, right? So they it's if there are common features across a cohort of patients that are assigned the same class, it would find those features. Now whether it would do well when a lot of labels are noisy, we don't quite know. There have been some experiments around that on toy data sets. Yeah.
Well, certainly confounders, and as you point out, the counterfactual issues become very complicated. Still, there might be a way to start to think about how to address the kappa values.
Yeah,
we'll talk about that. Other comments? But thank you very much. Those great answers.